Chapter 11, Section 1, Identifying the Substance of a Gene. Identify the clues that bacterial transformation yielded about the gene. Explain the role bacterial viruses played in identifying genetic material. And describe the role of DNA in heredity. About 100 years ago, biologists realized that to truly understand genetics, they first had to discover the chemical nature of the gene. In 1928, the British scientist Frederick Griffith was investigating how certain types of bacteria produce pneumonia, which is a serious lung disease. Griffith had isolated two very similar types of bacteria from mice. Both types grew very well in culture plates in Griffith's lab, but only one of them caused pneumonia. The disease-causing bacteria, the S-type, grew into smooth-edged colonies on culture plates, whereas the harmless bacteria, the R-type, produced colonies with rough edges. The difference in appearance made the two types easily to tell apart. When Griffith injected mice with disease-causing bacteria, the mice developed pneumonia and died. When he injected the other mice with the harmless bacteria, the mice remained healthy. Griffith wondered what made the first group of mice get pneumonia. Did the S-type bacteria produce a poison that made the mice sick? To find out, he ran some series of experiments. First, Griffith killed the S-type cells by heating them and injecting these heat-killed bacteria into laboratory mice. The mice survived, suggesting that the cause of pneumonia was not a poison from the disease-causing bacteria. In Griffith's next experiment, he mixed the heat-killed S-type bacteria with live, harmless bacteria from the R-type. He also injected this mixture into laboratory mice. By themselves, neither type of bacteria should have made the mice sick. To Griffith's surprise, however, many of the injected mice developed pneumonia and died. When he examined the lungs of these mice, he found them to be filled not with the harmless bacteria, but with the disease-causing bacteria. How could that happen if the S-type cells were dead? Somehow, the heat-killed bacteria passed their disease-causing abilities to the harmless bacteria. Griffith reasoned that mixing the two types of bacteria allowed a chemical factor to transfer from the heat-killed S-type cells into the living R-type cells. This chemical compound, he concluded, must contain information that changed harmless bacteria into disease-causing ones. He called this process transformation, because one type of bacteria, the harmless form, had been changed permanently into another, the disease-causing form. Because the ability to cause disease was inherited by the offspring of the transformed bacteria, Griffith concluded that the transforming factor had to be a gene. In 1944, a group of scientists at the Rockefeller Institute in New York decided to repeat Griffith's work. Led by Canadian biologist Oswald Avery, the scientists wanted to identify the molecule in the heat-killed bacteria that caused the transformation. Avery and his team extracted a mixture of various molecules from the heat-killed bacteria. They carefully treated the mixture with enzymes and destroyed proteins, lipids, carbohydrates, and the nucleic acid RNA. Transformation still occurred. Clearly, none of the destroyed molecules could have been responsible for the transformation. Avery's team repeated the experiments one more time. This time they used enzymes that broke down a compound called deoxyribonucleic acid, or DNA. When they destroyed the DNA in the mixture, transformation did not occur. Only one possible conclusion could explain the results. DNA was the transforming factor. By observing bacterial transformation, Avery and other scientists discovered that DNA stores and transmits genetic information from one generation of bacteria to the next. Avery's work provided evidence that genes were made of DNA. In 1952, the genetic importance of DNA was confirmed in a dramatic experiment performed by two American scientists, Alfred Hershey and Martha Chase. Hershey and Chase studied viruses, which are tiny non-living particles that can infect living cells. Their experiment was done with a bacteriophage, a type of virus that infects bacteria. These viruses attach to the surface of the bacterium and inject their genetic information into it. Once inside, the viral genes act to produce many new bacteriophages, which gradually destroy the bacterium. When the cell splits open, hundreds of new viruses burst out. Hershey and Chase designed an experiment to determine which part of the virus, its protein coat or its DNA core, entered the bacterial cell. To do this, the pair grew viruses in cultures containing radioactive isotopes of phosphorus-32 and sulfur-35. This was a clever strategy because proteins contain almost no phosphorus, and DNA contains no sulfur. Therefore, these isotopes could be used as markers, telling the scientists which molecules entered the bacteria. If they found radioactive S35 or sulfur-35 in the bacteria, it would mean that the viral genes were made of protein. If they found P32 or phosphorus-32, then the genes were made of DNA. They allowed the viruses to inject the bacteria and then tested them for radioactivity. The results showed that nearly all of the radioactivity in the bacteria was from the phosphorus-32, the marker found in DNA. Hershey and Chase concluded that the genetic material of a bacteriophage was indeed DNA, not protein. Hershey and Chase experiment with bacteriophages confirmed Avery's conclusion that DNA was genetic material. The DNA molecule must be capable of storing and copying genetic information, as well as putting the information to work in gene expression. The foremost job of DNA as the molecule of heredity is to store genetic information. The genes that make a flower purple must have the information to produce purple pigment. Genes control development, which means that the instructions that cause a single cell to develop into an oak tree, a sea urchin, or a dog must be all coded in DNA. Before a cell divides, it must make a complete copy of one of its gene. To many scientists, the most puzzling aspect of DNA was how it could be precisely copied without losing information. They could not even imagine how a molecule could copy itself, but that is exactly what DNA would have to do. Finally, cells must be able to take the information coded in DNA and put it to work. How does that happen? And what are the cellular mechanisms by which genes are expressed? The solutions to these many other puzzles would have to wait until the structures of DNA molecules were figured out.